I'm going to introduce Dr. Shetty and kind of give you a quick rundown on him. I can't read this biography and title. It's too long, Dr. Shetty's esteemed career. But I'll give you a quick introduction nonetheless. So Dr. Khalida Shetty is a professor of plant sciences and founding director of the Global Institute of Food Security and International Agriculture at NDSU. Prior to joining NDSU, Dr. Shetty was professor of food sciences at the University of Massachusetts for over 20 years. He earned his PhD in microbiology and MS in bacteriology at the University of Idaho, and he's later worked with the University of Guelph, the National Institute of Agrobiological Sciences in Japan, and within the U.S. State Department, as well, serving as a consultant. He's authored over 225 peer-reviewed journal publications, over 40 book chapters, and has been cited extensively globally. He has traveled over 50 countries and built strong research and education collaborations with institutions across the world, and mostly related to combating food insecurity and public health challenges on a global basis. So with all that experience, we're excited to have Dr. Shetty participate in our webinar series here at NCI, and today's being the Serial Innovators webinar series, and I'll turn it over to Dr. Shetty. So thank you so much for taking the time to be with us today. There you go.
Thank you, David, for your kind introduction. Happy to be here and present another angle about food barley and its health benefits. So like I do with more health benefits from food systems, I want to provide first a good rationale for why we do this and how the food systems globally are changing and the rationale behind that. And so as we target food barley for health targeted applications, I want to just provide sort of the emerging challenges. So we are a population of about 7.4 billion, about 9 billion, but at the end of 20, we'll probably reach about 10 billion, not 12 billion. But we have enough calories now for at least 12 billion on the planet, nearly double. We are 7.4 billion, but around enough calories for 12 billion. So if you look at that, then we have four big challenges that are currently before us. One is that we still have 1 billion people on the planet, about from the 7.3 who are hungry. But then we have a bigger challenge where we have a lot of this high refined calories that's available for 12 billion. And no wonder some of the fastest growing diseases are these comorbidity, preventable, non-communicable chronic diseases such as diabetes and its consequences like type 2 diabetes and cardiovascular health. So this is a bigger burden now than hunger itself. And sometimes they coexist in the same country and same region of the world. And therefore, these are the face of food security has completely changed since 2000, where we still have hunger, but we have this a bigger challenge from the excess calorie, where we have deficiency of micronutrients and other protective compounds that are not in the diet. So that's why we need to diversify our diet and keep those other fine components beyond just the major macronutrients. So that's a real theme that I want to address today and see how we use that rationale to redesign and relook at our crops beyond just the micronutrient model that was how we dealt with hunger, which is still a issue. We need the balance of macro and micronutrients, but we have this bigger challenge of preventable, non-communicable chronic diseases. And another important problem is that we need to address both these challenges with the climate rapidly changing. And interestingly, a lot of the resilience factors associated with health benefits are also some of the compounds that are needed to make the plants more resilient. So I will talk a little bit of that cross-tolerant health benefit and stress resilient, where the metabolism can overlap as we can channel the macronutrients towards these two functions. And a fourth important dimension is a lot of the antidotes for chronic disease, that's fruits and vegetables, they're highly perishable. So how do we manage these waste in a local setup and preserve these food and improve food safety will be an important dimension of food security globally. So there are other challenges too, but we see these four as a continuation of the current challenges that we face. So a lot of our solutions are linked around to these four challenges. So with this perspective, as I told you, we have still 1 billion people on a planet facing hunger, roughly about 1 billion. And then the higher number of facing the burden of chronic diseases. And this was very evident in the current COVID pandemic as well, because you can see the susceptibility of linked to comorbidity was higher. So in urban setups around the world, like in India or in the US or Latin America, there's an emerging perspective that comorbidities, particularly breakdown in the innate immunity linked to the hyper-processed calorie burden linked to chronic disease, they make our innate immunity much weaker. So you can see as the COVID ravaged around the urban environments of the world, that is an important factor we need to consider. Not just the metabolic syndrome breakdown towards chronic disease, but what is the consequence on our innate immunity? So this undernourishment, malnourishment, and excess nutrition, but the excess nutrition also having an important benefit for our better immunity. So these are important. So our public health challenge is very closely linked to food security as a result. And this has to be addressed with climate changing. But fundamental issue about food security is nutritional health, nutritional security that's health targeted. Not only to the major chronic diseases like type 2 diabetes and its consequences, which I will talk about today, but also towards the immunity, a balanced nutrition, a diverse diet that has better benefits for our innate immunity or broader immunity. Okay. So this is where our challenges lie, and how can we now retune the food? So now, even if you look at where COVID emerged and re-emerged, or the new strains emerging, you can see, South Asia suffering a lot, especially after the Delta strain, Europe. So in all of these countries in the circles, we don't know all the facets of what happened in some parts of the world. But if you see in major, you can see death rates between 1,000 to 2,000 per million. But beyond that, beyond the COVID itself, if you look at some of these circles, this is where a lot of the new hyper-processed, macronutrient-dense food-linked, diet-linked chronic diseases are emerging. Okay, particularly type 2 diabetes, which is the most rapidly growing disease on the planet. But the consequence of type 2 diabetes, it feeds into cardiovascular disease and all other disease, and ultimately cognitive breakdown. For example, Alzheimer's, it's really like a type 3 diabetes. It's really a sort of a brain-focused breakdown in sugar metabolism. And so they all, metabolically, they're all linked, but it starts with the glycemic consequences on the vascular health. So that's where I will focus on. And so how do we now take this new facet of global food and nutritional insecurity and retune the diversity of our food system, move away from just the major 10 crops we focus on to a wider array of crops. And that's where food barley comes in. It's already a food in some parts of the world, but it has a lot of benefits, just like the ancient grains we talked about. So in this widening platform of cereals, we can reach out to new range of health benefits. And so how the more wider we bring this, the more antidotes we can have. And what is exciting about this is, as we channel the photosynthesis towards energy metabolism, that energy metabolism is required for basic function. But what's exciting is when you channel the energy towards pentose phosphate pathway and make the anabolic link to making these specialized compounds that the plants use for stress and abiotic or biotic responses, many of those same compounds has relevance to health. Okay, so that's exciting. So essentially, we need to channel some of the carbon towards these protective elements for the plant. And so this is the foundation for some of the strategies for building abiotic stress resilience and defense mechanisms as we face climate change. How do we make them more resilient to withstand period of drought or period of salt stress or sudden flooding. And so essentially we have to move the carbon around. And the same, if you move those carbon towards secondary metabolism, some of those compounds can be channeled towards some of these foundations of some of the glycemic regulators or blood pressure regulators or inflammatory regulation. So that's a logic we are looking at, where we can shift the metabolic logic towards dual function of certain category of compounds, which have relevance both for climate resilience, and we can target it towards bioactive enrichment for functional foods or ingredients that have health relevance. So with this in mind, if we just look at some category of compounds, phenolics are one category. There are others. There are 8,000 phenolics that have been categorized, and different plants have used these various categories for the same metabolic regulation. They're just doing it in different ecologies or for different function, but they come from the same metabolic flux. So if we can rationalize that and start looking at that, then that's the way we are doing it. We can now look at those bioactives towards glycemic control, controlling the carbohydrate metabolism, so that you can slow it down, so you give the insulin more time to work its job and not suddenly require the demand from insulin. You slow it down. So there's a lot of these slowing down compounds in plants itself. So we need not go into the drugs. So a lot of the drug designs are based on this logic. But interestingly, many of these compounds can be had from food on a day-to-day basis. So that's the idea. So can we get that in food barley? Can we get it in emmer wheat or fruits and vegetables? And then if you look closely, there are other types of compounds that can regulate blood pressure. They're not as wide as the glycemic control compounds, but there's still a substantial amount we can find in the diet. So these food systems have compounds beyond just the macro and micronutrients. They have compounds that can have fine regulation of our metabolism. So that's what we are searching. Not only the major oxidative function around which all our energy metabolism works, we being eukaryotic aerobic organisms. So oxidative stress is a burden. So around oxidative stress, a lot of our energy metabolism and other functional metabolism work. But beyond that, there are compounds that can fine-tune digestive process, blood pressure control, insulin regulating functions. Okay, so a lot of them are similar compounds that the drug design have been focusing on. So our goal is, can we get this in the diet? And how do we then get the right variety and process it in a right way so that we retain those health benefits? Whether it's in cereal grains or legumes, which are rich in them, but also fruits and vegetables. There's lots of it in them. So how do we widen this with these metabolically, fine-tuning compounds? So that's the logic. So with that logic in mind, we have looked at cereal grains, and as you know, whole grains and its benefits. Particularly for chronic disease, reducing chronic disease risks, cholesterol regulation, the broad concept of antioxidant function, not just free radical scavenging, but how do they regulate the endogenous antioxidant enzyme response that couple to the energy metabolism. So then compounds that are anti-diabetic at the glycemic control level. So all of this are in the whole grain cereal. So if you can look at the grain itself and retain those compounds without just only using certain part of the grain, then we can retain those benefits. So barley, food barley, which has greater use in some parts of the world, can be one such opportunity, especially here as we try to balance between the beverage needs and the food grain needs. So if you look at that, if you see the hull versus hull and bran and germ, so each of those different parts have some of these unique, wonderful compounds. And so these are some areas of the world where it's used as feed versus food, but from a conceptual point of view, we are interested in these different parts where these unique health relevant compounds are found. And so how does growing and processing retain them? So that's the logic. So how do we do that? So in our case, we develop some of the tools to analyze them towards the metabolic basis for the health. So but both sides are important. Growing is important, the right variety is important. How do we process them is important. And how do we retain each of those components as we target them towards these health benefits is the goal. So as we go further, if you look at barley, both the endosperm, bran, and the germ or the embryo, all of them have these unique compounds. So if you see if we can do the whole grain milling, then we retain most of them. And now the question is, how do we target them and which of them have benefits for what health outcomes? So that's the goal. So can we stimulate them by sprouting? Can we stimulate them by fermenting? And if we ferment them, how fine should the grains be? Do we ferment them after sprouting? So ultimately, entire goal is how do you mobilize these compounds of interest towards health in the processing stage? And do we need biological processing, or you need other levels of processing? So that is very important. So as we look beyond this, in our particular case, we have been looking at the broad category of phenolics. These in nature, by nature, they have this broad function of the countering the free radicals at many layers, being oxidative organisms. This free radical scavenging potential emerges all the time because it's a critical part of the energy metabolism. So plants have developed these countermeasures to counter them. So can we capture some of that into our own dietary antidotes? And the answer is yes. So there are a lot of these, and literature is full of this. And we need to go beyond just the free radical scavenging itself to more fine regulation of our metabolism. For example, here the GABA is not a phenolic, but it's an amino acid derivative from glutamic acid. It's rich in barley. And this is one of the compound for, it's known as a yoga compound, right, for calming. So it's a very important neuromodulator. So a lot of these neuromodulators, gut digestive modulators, they all are rich in the grain. And so how do we now optimize them? Okay, so these are compound beyond just basic the macro and micronutrient. These are more fine compounds that do the fine metabolism, adjusting the a very specific stage of different types of digestive process or cognitive process. And so that's where we are looking at. For example, here, we want to look at barley as a functional food increase for the digestive process. So in the digestive process, the primary hyperglycemia is the primary stage as the burden of diabetes emerges. It's how do we control the glycemic load? So only when the glycemic load breaks down and the insulin ultimately cannot manage it, then you have the problem towards later stages of diabetes. But in the early stages, the drugs or many compounds that are there in the food, they slow down the digestive process. So in a hyper load of sugar, if you have all these fine-tune, compounds that can fine-tune the digestive process, then it gives the insulin better opportunity to work itself and manage how pancreas works in releasing the insulin. So many of these compounds can be found in the diet. And so we are looking at how we can sprout the barley to release them, how we can ferment the barley to release them, how can we use the beneficial bacteria to release them, how does the beneficial bacteria not only release the anti-diabetic and cardiovascular benefiting compounds, but also can they have the probiotic stimulating compounds, so which can have relevance for gut health or inhibiting ulcer bacteria. So those are some of the targets we have showing as proof of concept how these bioactive fine compounds can be targeted towards health. So moving down further, our general experimental model to develop barley-based functional food is following. We do identification extraction with the right species, cultivars, growing condition, environment, post-harvest stage, and processing. And from there, we have in vitro screening. In vitro screening is absolutely important because we can't do rat studies, or clinical studies are very expensive. So we need to screen a lot. So when we screen a lot, we have to do the close to important analogs with in vitro models. So we have a lot of these in vitro enzyme models, which is similar in the pharmaceutical. When they screen these pharmaceuticals, they first screen, on a wide scale before going into those few compounds that may end up in animal models or clinical models. So we have these in vitro assay models that are plenty. So based on that, we screen special varieties or cultivars or growing conditions or specific environmental effects or post-harvesting effects. So all of this we can screen by targeted in vitro approach, whether they are in vitro studies linked to digestive health or vascular health or cognitive health. And then the few extracts that are superior then can be rationalized for maybe some worm models, some whole cell models, okay, where we can see functional studies in a worm, or beyond that, you can then streamline it towards an animal model. Ultimately, that one special food ingredient that works, then we can target it towards, say, clinical studies. But before that, we need to go through this layers of sort of fine-tuning to get the health target. What is the health target in that particular species, in that particular processing stage? So that is the strategy. Therefore, rapid screening, we use the in vitro assay models that are very closely analogous to the in vivo models. And this strategy is important for wide analysis and further incorporating clinically relevant analysis. And then what can you then target into the diet? And the wonderful thing about food is they're already safe. We are not reinventing. We are already looking at existing food that we have already consumed for a long time, barley from 4,000 or 5,000 years ago. And so it's the safety is built into food, into the food system. And now we look at the special compounds in it. So we started with the some work with just the malting varieties or the beer varieties, and then we screened a wide before we went into the food. So I will not spend time. The whole purpose here is to look at the anti-glycemic function of some of these varieties, which have high, which have low. And so that provided the conceptual foundation for from the malting to go more towards the food grade. This is continuation of the work. We look at the broad category of antioxidant activity, but they are, they're not perfectly relevant. You need to also have in vivo responses. But anyway, they are an important indicator in the overall process. So we screen them, and then we see which of these phenolic compounds with antioxidant protection also have high anti-hyperglycemic relevance in terms of the in vitro assay that's related to the digestive or the glycemic control in our own body. So we have the enzyme equivalent to that. We usually get a porcine or yeast. They're close to similar. That's how even in the pharmaceutical industry they have the similar logic of the enzyme targets. And so with these high phenolic and sort of phenolic profiled compounds, we look at the general antioxidant assay, which doesn't say much. But beyond that, what says a lot is how is this digestive enzymes regulated? Can they be inhibited? To what extent are they inhibited? At what stage are they inhibited? What variety inhibits it? And so you can see those arrows. What stands out in terms of the digestive inhibitors, we pick them out. Which variety, which stage. So here you can see the anti-hyperglycemic or sugar lowering effects of sprouted barley grain in vitro assays. So these in vitro assays will inform which variety or which stage of processing are superior. And then we couple it with sprouting and other strategies like using beneficial bacteria, which does two things. One, the bacteria itself biotransforms the matrix towards useful compounds. And second, those beneficial bacteria in themselves can be probiotics. So we can couple these microbiome into the processing, but also couple the microbiome into health benefits. So based on this, we see further how are these anti-diabetic compounds or probiotic stimulating compounds or antimicrobial compounds against certain pathogens can be screened. And therefore, we profile them, we ferment them, we see which stage they are optimized. Are they stable during fermentation? So improvements in stability and mobilization of phenolics. And here we've used a sort of a 10 culture kefir combination to release these functional compounds. And so you can have other microbial targets as well, as you see fit for different plant substrates. And so with this fermentation, we can see that many of these alpha amylase and alpha or even alpha glucosidase inhibitors, or that's related to the anti-hyperglycemic function, they can be modulated and stabilized by fermentation. Okay, and alpha amylase is the first step where the starch is broken down into smaller units before alpha glucosidase helps in the uptake. So when these two steps are properly modulated, then the insulin demand emerges, right? So when there's glucose that then has to be managed to be distributed to each of those cells. So that's when the insulin comes in. But just before the insulin, can you slow these early digestive steps? And what are those compounds? Can you do it by fermentation? Can you, which stage of processing, which variety? So with that in mind, we've screened, and we have also found some of them have inhibitory effects of the ulcer bacteria. This ulcer bacteria is very interesting. It's an aerobe, it's a microaerobe. That means to some part of its metabolism, it still needs oxygen. But what's exciting is the lactic acid bacteria do not need oxygen for energy. So the same compounds that are supporting the lactic acid bacteria, they don't do anything to the lactic acid bacteria or to our host cells, because we are a eukaryotic mitochondrial based system. But H. pylori is exposed because they have an oxygen dependent energy metabolism right on the outer, just above their the membrane linked to their peptidoglycan. And therefore, compounds that can be inhibitory to the pathogen is not inhibitory to the host cell or the lactic acid bacteria, because they have different functional responses to the compounds. So here it's exciting that beneficial compounds that do not do anything to beneficial bacteria can inhibit the ulcer bacteria. So this is very useful. So we have further looked at some commercial they are barley tea from Japan and South Korea, roasted barley. They're not coffee itself, but made like coffee or made like tea. Okay, just names. But they're all made from barley. So we looked at, okay, what are these commercial, existing products? What are they? What is their soluble phenolic content? Do they have the anti-glycemic? What is their phenolic profile? Then do they have when you process, when you use them in the time how you consume them, like in barley tea or barley coffee, what is the antioxidant activity when it's released and at what time? So how much of the tea bag you need? Is 3 minutes, 6 minutes? When is it optimally released? And if so, does it translate into the anti-glycemic function? So if by drinking barley tea or barley coffee, can you have the digestive inhibitors, right? And so instead of a drug, you drink barley tea or barley coffee, right? So that's the idea. And some of them are okay, some of them are not. So what does this mean? That the barley tea and barley coffee need to be better rationalized with the right variety, right processing stage, right? So this was just, they probably took any variety, okay? So when you now reinvent this, you need a better superior variety. So that's how our research can inform how this better barley tea or better barley coffee can be made, okay? So that's the logic. And so we continue with that for different anti-glycemic alpha glucosidase and alpha amylase because they go hand in hand. One releases the starch to the smaller units to maltose, maltotriose, and glucose. And glucose then is taken up by the enterocytes before the glycine, insulin responds to that that glucose load. Okay, so you can manage those alpha amylase, alpha glucosidase steps with dietary inhibitors. Then you give the insulin more time, more time to work, right? So in this era of high sugar rush, can you slow it down? Can you slow down, right? So that slowing down can be done in the diet very safely and effectively. We don't have to pop pills for that, which is what happens ultimately when the sugar is not managed. You get metformin first, and then there are other type of drugs as the system breaks down, right? So if you go and have the early stage of diabetes, they recommend metformin, which is, which actually just moves it slows down, but actually it moves the glucose into the liver and sort of keeps it in the liver. And then from the liver, the fatty acid metabolism takes over, but ultimately they sort of feed back into the insulin loop, the fat that goes into the liver. So these metabolism, they are very interesting because the body is built to build an initial safety net for you. So that's why they sort of parcel it off to the liver, right? But ultimately, the liver can become fatty liver when it gets diseased, right? So all of this metabolism, can we program that into the food as best as we can, and how do we screen them? So that's how we are looking at this. Here's a very exciting where the proliferation of beneficial bacteria. I told you it can inhibit ulcer bacteria, but it allows proliferation of beneficial bacteria. Why? Because the beneficial bacteria do not use oxygen for energy. Ulcer bacteria uses oxygen for energy. So if you can capture that oxygen coupling step and inhibit that, then you support the good bacteria and you inhibit the bad bacteria. So that's the logic between supporting beneficial bacteria like bifidobacteria versus the ulcer bacteria, which can be inhibited. So in summary, food barley is a rich source of phenolic bioactives. There are other compounds too, which we're interested in with high antioxidant potential, which doesn't say much, but we have to translate that into in vitro antioxidant enzyme response function. But more importantly, can it modulate the activity of key enzymes associated with digestive process, which can then be targeted towards the anti-diabetic benefits, especially in the early stage of the disease, where diet can be a good antidote to counter it with exercise before we even get into a serious drug regimen. Food can support that? And the answer is yes. How can we redesign the food, both in a fermented stage by sprouts or tea or any form? And can we rationalize all of that in a functional level in the design, all the way from the right variety to the processing to the different stages, right? And so food science is very exciting because you can channel all of these health compounds from all the way to soil health to human health. And so this is where the research is heading. So we need to look at food security beyond just macronutrients and micronutrients, but also this fine regulation of our metabolism, because that's where a lot of our health breakdown is occurring. And here is an example. Last month, I showed you emmer wheat, this ancient wheat, have potential. So as we bred away and made macronutrient dense crops, we bred them away. We need to go back to them and capture some of those health benefits and rethink how we have our food and have all those ingredients still retained. And how does all the technology we have developed, even the stone mill, you said, can we use the stone mill to keep everything good, So it's an exciting time for research. So we need to move some of the best extracts from our screening into animal-based models. So until you prove it in animal-based models, it won't go to the clinical level. But we can't channel everything that we study towards animal. Only some of the best ones can move to animal. And we need to see in different ecologies, how are these bioactives working, which variety, is it high mountains or in the plains here in North Dakota? Is it the Western North Dakota where you have more dry land? Because sometimes dry, you may lose yield, but it may be better for bioactive compounds. So some of these plants may be more suitable for these specialty ingredients, more in the dry land if you can compromise some yield. So the stress may be good, and you can compromise some yield towards some of the health benefit compounds. So how are different processing strategies, relevant in this? And so ultimately, how do we get this barley-based functional foods or ingredients at various aspects of our health need, particularly early stage of the disease. So we are interested in building this sustainable food system, both for climate resilience and health. And of course, through NCI, it's always a pleasure to work with NCI as they build these global partnerships. We also try to work with them, and we have also community engagement at many levels. And we have an annual Food for Health conference, which traditionally has been here in Fargo, but now 1 year we have it in Fargo. Another year we have it in Bali, Indonesia, so that it's closer to the market, right? And so when we have it closer to the market, that's where a lot of the high value exports from here can be targeted. And Asia is the market where 65% of the population of the world is. And so if we take our research innovation and through our community engagement, our goal is how do we add value to our local growers, working with NCI and research within our institution, and then disseminate this and engage through our workshops and conferences. Because we also publish a lot, so we'll be happy to send some of our papers. And if you need this presentation, I can make a PDF copy and can be available to all, whoever requests. And I'll be happy to answer any questions. So thank you. And I think we have a lot of potential with food barley and where it can be taken. And I've given you sort of the rationale how we can go through that, from growing valuable varieties to all the way to processing into the food design. So thank you, and I will be happy to answer any questions as they come.
Very good. Oh, we appreciate it. Yeah, so if anybody's got any questions, feel free to send them through the chat. You've got a chat box in the bottom, so we'll take any questions. I've got a number of them. So thank you, Dr. Shetty. I think for me, the first point is that I'm excited to see your excitement in general with the subject. So talking about food health and tying in, of course, to global food insecurity in this case, as you talked about earlier. So it's really exciting to see your passion. So good on that one. I guess so. Yeah, I'm going to ask a couple questions. I've got a number, and so I'm thinking like an agronomist. I'm thinking like an economist. I'm thinking like a farmer.
Sure.
So it's maybe a little less on the food science, but it's really interesting to see how the impact that this has basically on a food system or a supply system. So a couple questions that I've got. My background in plant breeding, and I'm thinking about these micronutrients from a lens of, say, a plant breeder, and in that respect. The challenge of breeding for yield and breeding for pest resistance and environmental stresses and things like that. And when you add in, these micronutrients and types of things that, of course, are beneficial on the food side, how does a plant breeder interact with that kind of information, and how likely, maybe if you can understand?
Very good question. So many of these compound fine regulation can happen at the gate where the carbon flow emerges versus late steps. So a lot of the time, we need initially some simple link to how breeder can, because breeding is a longer process versus, say, you pick up a gene of interest and you can multiply. But that's, there's a gap in that. But just let's take breeding strategies. I understand it's a long, drawn-out process, and rightfully so, as you screen through different generations. And so one would be simpler gateway genes. So a lot of this carbon flux moves into the secondary pathway, right? So some of these gateway genes related to pentose phosphate pathway. So that could be the first line where we help, a metabolic understanding to some single gene targets that are major control points, which moves the flux towards these compounds. and then build from there. And so keeping it simple, right? So first look at it. So we know that these gateway pathways take it towards these health compounds. We know these gateway pathways take it towards these stress compounds, because nature doesn't reinvent. It takes common threads and sort of responds to the same environment. So we find such logic and pick such genes of interest. So that'll be the first simple approach.
Yeah, I would imagine it'll be a long step process too, right? Once you identify the compounds of interest and how they truly affect, downstream
Right.
health quality.
Yeah, it's better to start with upstream and take those upstream critical control. And so one example is pentose phosphate pathway. It's the number one pathway that moves all compounds to all these stress protective pathway. So hanging around pentose phosphate pathway itself could give the markers for the plant breeders. Right.
Well, I know we've got a number of folks listening that are part of other grain organizations as well, aside from barley. So I guess one of the questions I've had maybe is, I know you're working and talking about barley today, but it's from across all cereal grains or ancient grains rather as well. Has there been as much investigation?
Oh, very good. Very good question, because just last month I gave a presentation on emmer wheat. So this emmer wheat has its own history, that it came with the Germans from Russia, right, in the last 1885, around that time. But it still was the main emmer wheat that was maintained by the immigrants who came here. And so we have few growers who still maintain that. So we have been studying that for the health benefits. And in fact, we've just published a couple of papers on that through one of my PhD students. So this ancient emmer wheat has its benefits, and we compared it to the couple of regular cultivars versus that. And so it has potential beyond that. Then we have collaborations from Africa and other places where we look at their local grains. So we are published with colored corn from Andes, right? Especially high Andes. And because they use this purple corn and this amazing corn that goes into chicha. So we have studied this chicha fermentation and how some of these bioactives. So it can be tailored to different contexts of different environments. So we've been studying that. We've been looking at, we just had a PhD student who just completed the whole study on sweet potato. So as you know, sweet potato is becoming an emerging nutrient-rich crop for world food security. So we looked at these different purple and yellow and seeing some of these health benefits. So her whole thesis was on sweet potato.
Well, I've got a number of questions I want to follow up with that.
Sure.
Before I do, we do have a question from online, and they're asking about, so we talk about different cereal grains. In this case, the question is, are there significant differences, nutritional differences between malt barley or hulless food barley varieties, and particularly after you process them?
Yeah, the answer, I don't have direct evidence to point out, but the answer is very, from whatever we have done, yes, quite likely. I can't tease them out because we have done some barley variety screenings, and also a food variety, few food barley varieties that we have done. And they are in the similar range, but I can't sort of right away tell you how different they are. But the same phenolic flux that goes into the malting versus food, they are related. There may be other variations in other compounds, but in some of the compounds we are targeting for glycemic index, they are some common factors in the malt itself before it's fermented to a beer. And so, like, the Icelandic, Iceland has a winter malt before it's fermented, like a drink. So that's pretty rich in some of these compounds. So, but they take the same malt towards beer, but they just have the 48-hour malt as a drink, as a Christmas drink.
Sounds good. Yeah. We'll have to bring that here. I'm German from Russia, so I guess I'll try some Icelandic things as well. Very good. Well, and on that point, you talked a lot about fermentation and sprouting and such. And I know when it comes to processing, and one of the things that we do at NCI is talk about the effect of processing. So in that respect, fermentation alone and sprouting and other things, but related, and I guess my question is about heat.
Yes.
You talk about enzyme and enzyme activity, which of course is heat, can be heat driven. So you think about the processing.
Yes, very good. So some of these compounds are tucked away in the vacuole, and vacuole, as you know, in the plant is very unique and big. And so it's really a sequestration defense strategy for the plant, where it takes some of these compounds that can not go into the normal phylum, phloem loading, right? And it tucks it away. And what we are seeing is sometimes when you heat them, right, what may emerge out of the vacuole may take its time, it may be beneficial. And there are other cases where in the vascular system it's loaded, right, it can be destroyed. So then we may have to optimize them as you take it through different processing. Say you have an ingredient end product, some flour of some kind. So we'll have to optimize, okay, what happens when you sprout and ferment, or when you make a flour out of the different stages? Does the heat destroy it? And the answer, in some cases, it may destroy 20%. In the other cases, they make a big compounds. We've seen it in the case of fava bean. There's certain L-dopa that's linked to cognitive function. And we have evidence that you give a small microwave dose to the sprout, it actually increases the compound. And that's because we rationalize it's hidden up in the vacuole, and then this heating throws it out, which otherwise you may have a harder time pulling it out by regular fermentation or other mechanisms. So it varies on the situation.
So a fair bit of study, you think about like coffee, you mentioned a couple of examples on coffee, which obviously would be.
They may have lost it.
Yeah. So it could be even better.
Correct.
Depending on how the coffee is made.
So maybe there's a variety, they've lost it all. So then maybe we have to go to some high variety and lose some of it.
Well, I'm a fan of coffee, and chicory coffee, notwithstanding, I think I'll try probably more than that. So that's good. I'm going to switch gears a little bit and talk about some of your presentation related to diabetes. And I have to be honest, one of the first thoughts I had with the increase of diabetes, of course, in the United States and in other developed countries, is it a question about calorie intake or calorie quality?
It's calorie quality and intake. And it's a lot to do with the soluble carbohydrates, which can come both from easily digestible starch to sugar, both. Because sugar ultimately becomes glucose, right, in our vascular system. So it's from both. And so people have been blaming fat. Fat is less to blame than highly soluble carbohydrates. So it's really our glycemic control is the big culprit. And we still need sugar, yeah. But how do we meter that to the right level, coupled to our, how we function in every day, right? And so it's really the glycemic load. And the first defense of our plant, when we have too much of it, our body is also reacting. One, ultimately it may break down, but then it first makes fat. So excess carbohydrates are put into liver, right? That's a normal. And that's how metformin works too. They quickly parcel it off the liver, but ultimately liver becomes fatty liver, right? So you can have fatty liver from sugar or from alcohol. Both feed back into. And so that's where, how we slow it down at every stage. So even pharmacy is approaching it that way, but if we can sort of slow it down with daily diet, that would be better approach, right?
Yeah, exactly. No, it's interesting because they talk about diabetes and the effect of whole grains on diabetes, and yet in the United States, it's not a matter of food availability, right?
Correct.
We have plenty of food in this country.
Yeah. We have, in the US, for example, we have, I'm not saying all take 4,000 calories. Each citizen of US has availability of 4,000 calories. I'm not saying all take 4,000 calories, but we need 2,000. But how is that 2,000? It should be, can we have it in a diverse whole grain form, right? Right.
So on that part, it seems like there's a lot of information you talked about available, and more research should be done, of course.
Sure.
But I think the industry is quite aware already that whole grains need to be part of our diet for all these reasons you discussed. And so what is the medical community doing?
Very good.
Are they paying attention to the whole foods approach?
Now, For example, now your Mayo Clinic and the Cleveland Clinic, they all getting into because it's the collapse. They are seeing the collapse, and so even insurance companies are starting to think about that, right? Kaiser and a few others, right? And so ultimately, it's and also the biggest challenges is the curriculum of the medical students. So traditionally, they had this, 3 credit simple nutrition course, which still tells you macronutrient 2,000 calories. The same thing, right? So that's the information. So it doesn't hit the thinking when they meet. So 85% of our healthcare costs are preventable chronic disease, but they only take one course in nutrition, not in food science. Oh. Right. So there's so the medical training too is, I think, slowly changing in some parts of the world.
Right? So your conference, like your Food for Health conference and things like that, I mean, you talked about the folks that are involved in the message there. Are you reaching out to them?
Oh, yes, yes. So in all our, yes, we bring the medical people into the picture and have them. And so in some cases we are ignorant, in some cases they are ignorant. So we sort of both cover our ignorance.
That's the point about communicating.
Right.
Very good. Well, that makes a lot of sense. So I think you're right. And in the food development, where we talk about food product development, one of the hang-ups or the challenges of getting whole grains into the diet is people say it doesn't taste as good, where you have the phenolic compounds, it's bitter, and maybe some folks don't like that mouthfeel of a whole grain.
But I think that even then we can make it, we take all the factors that need to make the food better. If it's taste, okay, now how do we put the health and the taste, right? So I think we need to just work towards a problem.
Yeah, exactly. Well, and that's exactly what, like Northern Crops Institute and the groups that we work with, that's our goal, right, is to get those crops utilized. And so how do we best do that in a way, whether it's fermentation, which definitely changes the flavor, things like that could help with those roadblocks to getting more whole grains in the diet.
And in some case may taste better. So it's like many of these foods, we just saw them recently, right? So we just build acquired taste or rechange.
Very good. Well, we're running out of time. If anybody's got any questions, feel free to get them in quick, or I know they can reach Dr. Shetty directly. If you don't have his email address, for sure, contact Northern Crops Institute, and we'll be sure to pass email addresses on so we can answer some more of these questions if you have them. So I guess the last question I'm going to have for you, and maybe it's one you've already talked about briefly, but as far as isolating some of these compounds, is there ways of using them in a way that fortifies other foods that are entirely
so their benefits in some contexts and no benefits in the other, because many a time it's the synergistic regulation that's important. So we have some evidence to show that if you have a compound in a whole form, and then as you start purifying, you lose function. And so that means when you're in the whole stage, that particular glycemic regulation is higher. And we still yet to figure out that completely. How do they work synergistically together? In some metabolic science, it makes sense because you have common control and each compound may have different dose of response, and different plants make the same compound, different compounds for the same function because they're in different ecology. So there is some fine tuning, and what we are seeing in some studies that as you strip them down, you lose function, especially these gross metabolic function, right? And so they may work together. And so that's actually a big challenge, because a lot of the drug discovery is single target approach. But we need to go to a more metabolic approach because that's the way a body works. But that's challenging because what is that control point you hook on to screen them and see how these synergies work, right?
I'm thinking again about the developing the markets. Can you pull something out of the barley and then?
There are some. There are marketable situations for single compounds to be added, and there are situations where you need a whole platform, right?
I'm the guy trying to add value, right?
Yeah, maybe you want a drink and then suddenly you see some benefits of a compound. It can be pulled into a drink. I think it's just, can you make anything healthy by adding one compound or 20 compounds or whole extract?
Well, very good. Well, we appreciate that. We just had a question came in. We'll see if we got enough time to answer it real quickly. With an increase in whole grains, we'll see an increase in phytic acid consumption.
Yeah.
If the product hasn't gone through a fermentation process, aren't the negative impacts?
Yeah. I agree. But then we'll have to find a way to reduce the phytic acid. Yeah, that will be a issue in some situations, the calcium uptake, and it will have a downside. Yeah, so nothing is black and white.
Yep. So there's much work to do.
Right.
So, yeah. Well, it's another reminder that we need food scientists and biochemists in agriculture. Right? So we definitely need that. All right, well, we're going to wrap it up. We're up against the time, so I'm sure we could probably continue to talk. Dr. Shetty, we appreciate your time today. Appreciate your expertise and, again, your passion on this particular subject. For anybody that wants more information, feel free again to reach out to Dr. Shetty or Northern Crops Institute. Our website again is northern-crops.com. If you want to send us more questions or comments, and look forward to more of our Cereal Innovators program coming for the future, as well as our other webinars. So thanks for everybody for joining today. Thanks for your time, and we wish you a good day. Thank you, Dr. Shetty.
Thank you.